Bismillahirrahmanirrahim. Assalamu alaikum. Welcome back to Biosciences with Shatrin Sawati. And today I will continue my lecture about the carbohydrates. Yesterday I delivered a lecture about the monosaccharides and stereoisomerism, and I will start where I left it. So today I will discuss about the oligosaccharides. I have discussed a little bit in my previous lecture about the oligosaccharides. Oligosaccharides are the second class of the carbohydrates, and the most important oligosaccharides are the disaccharides. Disaccharides mean the sugar which contain two monosaccharide molecules. So oligosaccharides, as we know, that contain from 3 to 9 molecules of the monosaccharides. So the examples of the disaccharide, most common lactose. We are familiar with the sucrose. We are familiar with the maltose. Lactose is found in the milk, and this lactose is made up of the by the reaction of the glucose with galactose. And the reaction is known as condensation reaction, in which water molecule releases when two molecule monosaccharides, glucose and galactose, when react with each other, produces the lactose. Similarly, sucrose, by condensation of the glucose and fructose, produces the sucrose, and that sucrose is commonly known as a table sugar. And the maltose, which is found in the malt, and the maltose is produced by the chemical reaction between the glucose and glucose. So these two monosaccharides, glucose, galactose, glucose, fructose, glucose, react with each other. As you can see here, the bond. This bond is known as glycosidic bond. This bond is known as glycosidic bond. Now, glycosidic bond may be 1,4, maybe 1,6. I will show you. Now, here you can see in case of the maltose and lactose, there is a 1,4 glycosidic linkage. Means 1,4 linkage means the here you can see here, this is the carbon number 1 of the. This is maltose, and maltose is made up of two sugar molecule, two glucose. So this is the carbon number 1 of one glucose, and this carbon number 1 bond with the carbon number 4 of the other monosaccharide, other glucose. Monosaccharides mean I'm talking about the glucose. So the bond is produced by condensation, and this bond is 1,4 glycosidic bond. As you can see here, this is the one glucose molecule, and we know that at this point, at carbon number 1, OH hydroxyl line is present, hydroxyl group is present, and here it's the same case, hydroxyl group is present. So during condensation, what will happen? Hydrogen from this and OH group from this monosaccharide or glucose releases and produces the one water molecule, because this is condensation. And these two monosaccharides, two glucose molecule, react with each other and form a bond which is known as the glycosidic bond. Is it clear? So same in the case in lactose, same in the case of sucrose. You can see the glycosidic bond, the bond which is formed between the two monosaccharides, or two glucose, or two fructose, or glucose, or galactose in case of the disaccharides. You will see the 1,4 and 1,6 glycosidic bond or glycosidic linkage in case of the polysaccharides. So these are the linkages, these are the bonds which are produced between the monosaccharides, and then monosaccharide produces the disaccharides or oligosaccharides. Is it clear? Okay. Now, polysaccharide. Poly means many. Saccharide, saccharons, sugar. Many monosaccharide units react with each other and produces the polysaccharide. The important polysaccharide in nature are number 1, glycogen, starch, cellulose. Glycogen, we are familiar, this is the animal starch. Starch, plant starch, which is present in the plant. Cellulose, we know that the structural component of all the plant cells, cell wall, so it is present there. And the same case, these polysaccharides, these all monosaccharides unit link with each other during condensation and produces the glycosidic linkage, as in the case of the disaccharide. Okay, first of all, I'm going to discuss about the glycogen and the starch. Glycogen, it is a long and branched polysaccharide. Glucose polymer, it is also known as glucose because it is made up of the glucose units. And glucose is a monosaccharide that human and animals store in the liver and the skeletal muscles. So it is the, you can say, the store energy, store which utilized during many other processes for the production of the energy. So starch, it is available in plants. It is the main carbohydrate of the diet. We are taking starch in different forms, in different food items, in our daily routine. So the concept is clear about the glycogens and the starch. These are what? These are polysaccharides, are glucose polymers. Okay, now starch. Starch, which I have discussed, that is present in the what? In plants. Starch is found in plants, and starch exists in two forms. You can say that two layers of the starch. One is known as the amylose, other is known as the amylopectin. Now, what is amylose? This is the outer layer of the starch granule. What is amylose? This is the outer layer of the starch granule. Molecules are linear. Must keep it in your mind, the difference between, if you have to solve any MCQs related to the amylose and myelopectin, must keep it in your amylose is the straight chain, linear molecule with no branches. But the amylopectin, this is the inner layer of the starch granule. Outer layer is the starch granule, outer layer amylose, inner layer amylopectin, and this inner layer is long and branched. But amylose is what? This is the straight. I will show you here. You can see here the difference between the amylose and amylopectin. This is amylose, in which the sugar molecules join with each other by the 1,4 glycosidic bonds. So in this case, this is the amylopectin. This is the outer layer of the, which is straight chain. But this is amylose, and this is what? This is amylopectin, which is branched. Two types of the glycosidic linkage present here. 1,4 as well as 1,6. And now the concept about the 1,4 and 1,6 clear upon you people, that the 1,4 and 1,6 is related to the number of the carbonate at which the bond is produced. Is it clear? So this is amylose, which is the straight chain, and the other one is the myelopectin. This is the branch chain. This is the difference between the amylose and amylopectin. Okay. Now you can see here alpha 1,4 glycosidic bonds and beta 1,4 glycosidic bonds. You can see here the difference between the two, alpha form and beta form. Okay. Now structure of the glycogens and amylopectin of starch. Very interesting thing about the glycogen and amylopectin is that glycogen and amylopectin both exist in branch form. Amylose exists in straight chain form, linear form, but glycogen and amylopectin of the starch are branch chain. Polysaccharide made from a alpha D glucose. Now I have given you the concept of D glucose, mean dextrorotatory glucose. Glucose molecules are bounded by what? They are bound by 1,4 linkages as well as 1,6 linkages. The concept clear now? Okay, as you can see here, the glycogen molecule with branch structure Both types of the 1,4 glycosidic linkage and 1,6 glycosidic linkage exist in the branch Amylopectin as well as the glycogen animal starch. Okay. Now complex carbohydrates. Carbohydrates exist in complex form, like in nucleic acid in the form of ribose sugar, which is 5-carbon sugar, is present in the nucleic acid, both RNA and DNA. In case of the DNA, ribose is deoxyribose, and I will give you the concept of deoxyribose sugar and oxyribose sugar. And in case of the DNA, this is deoxyribose, and in case of the RNA, this is oxyribose. So ribose sugar, which is the part of the nucleic acid and form the complex carbohydrate. Similarly, proteins combine with glucose units and form the glycoproteins, which are present in the all the cell membranes and also the different organelle membrane, nuclear membrane, glycoproteins, and perform different functions, as I have discussed in my lecture about the cell membrane structure and cell membrane. So in case of when they combine with the lipids, they produces the glycolipids. So there is no need to explain about the glycolipids and glycoprotein. I have discussed in detail in my previous lecture about the glycolipids and so must watch all these lectures. Okay, ji. Reducing sugars. These are the, if the oxygen on the, what are basically first the reducing sugars. If the oxygen on the anomeric carbon of a sugar is not attached, must keep it in your mind, oxygen on the anomeric carbon, I have given you the concept about the anomeric carbon, which is the carbon of the what functional group, like aldehyde group or keto group, functional group, which are attached to the sugar, or you can say that the monosaccharides, glucose units. So glucose, fructose, galactose, all. So what will happen if the oxygen on the anomeric carbon of the sugar is not attached to any other structure? If not attached to any other structure, the sugar can act as a reducing agent and is termed a reducing sugar. Is it clear? Act as a reducer and is known as or termed as reducing sugar. What will happen in case of the reducing sugar? Oxygen on the anomeric carbon of the sugar is not attached to any other structure. Okay. Medical importance, these sugars can react with the chromogenic reagents as Benedict or filling solution, causing the reagent to be reduced and colored. Is it clear? And this is used in medicine for diagnosis of presence of glucose in the urine. Is it clear to you? What are reducing sugar? Why they are called reducing sugar? Because of the oxygen on the anomeric carbon. Now, plant cellulose, that is very important. Plant cellulose, as you can see here, the structure of the cell wall. If you see, this is the cell wall, and cell wall, you know that made up of the primary cell wall and secondary cell wall and the middle lamella, which is present between the two adjacent cells. So cell wall is made up of the cellulose, and cellulose are arranged in a crisscross arrangement. As you can see here, the cellulose arranged in a crisscross in order to provide the strength to the plant cell. So cellulose is a molecule consisting of hundreds and sometimes even thousands of the carbon, hydrogen and oxygen atom. Cellulose is the main substance in the wall of plant cells, helping plants to remain stiff and upright. Human cannot digest cellulose because human do not possess have cellulase enzyme, but it is important in the diet as a fibers. And this is the chemical formula C6H10O5 because the number is start from the hundreds and even in the thousands. Is it clear? Okay, now here you can see this is the plant cell wall in which the cellulose arranged in a crisscross arrangement, and this is just one fiber, which is the microfibril, and this is the cellulose molecule, which is further made up of the monosaccharide sugars. Is it clear? So this is the most important polysaccharide which is in the nature and found in the cell wall of the plants. Okay, importance of carbohydrate. Carbohydrates are initially synthesized in plants by photosynthesis, as we are familiar. Carbohydrates are important for providing energy, store energy in the form of the glycogens, provide energy by the breakdown of the glucose molecule, and supply carbon for synthesis of other compounds like all the proteins, lipids, and many more in the cell, form structure component in the cells and the tissues in the form of the cellulose, in the form of the starch, in the form of the glycogens, in the form of the glycolipids, glycoproteins. So all these are the importance of the carbohydrate. I hope so the lecture will be clear upon you people. Must watch my first part about the carbohydrate and then this second part. Like my videos, share my videos. Thank you very much. I will be back tomorrow.